welcome Weihsueh Chiu here. Come back from being here for a working group meeting that we had about 2.5 years ago, and this is our third attempt to actually have him come and give a seminar. The weather interfered once, and then the second time that we tried, neither Eric nor I was going to be here, so then we've now made a success of the visit. So I know Weihsueh from participations on National Academy of Science committees. In fact, we were together in DC earlier this week for a workshop on data integration at the National Academy of Sciences, and is a major contributor to that effort. At that workshop earlier in the week, someone was introducing a speaker and said, well, this is probably the only speaker you'll see who has a physics background, and then, Weihsueh saying, no. So he's got an undergraduate degree in physics and a graduate degree in astrophysics. Is that?
I was in physics, but it was actually in the physics department. But my advisor
was also in policy.
Right.
Yeah.
But my advisor was actually in astrophysics.
In astrophysics. Okay. And so at the same time he was doing that, he was also getting a certificate in science, technology, and environmental policy. This is at Princeton. So started at Harvard, went to Princeton. Reasonable institutions, not so good as Brown, but it's okay. And then went from that academic environment to government work, initially at the General Accounting Office in special projects. It sounds sort of like
special studies and evaluation.
Yeah, it sounds like CIA stuff, but I think it had to do with numbers, since it's the General Accounting Office. And then from there to the US EPA, and spent quite a bit of time at the US EPA and was the lead on the trichloroethylene IRIS report from TCU, which was a huge effort. I've read parts of it. It's extremely repetitive, but it's still 1,175 pages long or something like that. And it was sort of the definitive judgment on and setting of action levels for TCE in the United States, which was an effort that probably took you at least 5 years, probably more.
Yeah, I became the lead for that in 2003.
And then the report is 2012, right? Yeah, that's a really long time. So then 3 years ago, Weihsueh went from that government setting to an academic setting, Texas A&M, and a professor position at Texas A&M. And Texas A&M is one of the new Superfund research program sites just awarded this past year. So again, he's been a critical piece of that and will be a critical piece of their success going forward. Welcome. We're very much looking forward to your talk. Let me get the lights down for you.
Okay, thanks very much again for that kind introduction. Happy to be here finally. It did snow a little bit yesterday, I guess, but not enough to prevent me from being able to get here. So everyone see that already? So I'm going to talk about the critical issue of addressing uncertainty and population variability in risk assessment. I'll go talk about some of the challenges and opportunities I see in addressing this issue. First, my conflict of interest. I don't have any conflicts of interest. At this NAS workshop earlier this week, one of the speakers had in 10-point font 50 different firms that or pharmaceutical companies, all sorts of commercial enterprises that he's been involved in, and then he said, I don't blame you if you don't believe anything I say. But fortunately, I don't, and hopefully you will believe what I say. All right. So I was told, because there's a mix of sort of toxicologists and engineers, to give a little bit of background on toxicological risk assessment, segue into why we care about population variability and susceptibility and how important that is. And then a lot of my talk will be talking about some emerging approaches, both in vivo, in vitro, and in silico, to address some of these challenges. And then a few final thoughts about risk characterization in sort of a new world in which we have better population variability and susceptibility information. So risk assessment really goes back to the Red Book. How many of you are familiar with this particular National Academy report? Only Kim. So this is risk assessment in the federal government, managing the process, and it laid out sort of 3 different related areas. One is research, which is what people here in academia do. The second is risk assessment, which is taking that research and trying to sort of synthesize that into some conclusions as to whether things are hazardous, what the relationship between some response and exposure is. And then risk management, which is actually deciding to do something about what we're finding. So most of what I'll focus on today is over here, sort of the relationship between research and risk assessment. And I'll even though I do have a policy certificate, I'm not going to go a bit much into that, but I'm happy to talk about that if you have questions. So, for better or for worse, every time I go to Society of Toxicology meeting, people quote Paracelsus, sort of a late Middle Ages, like sort of physician-ish person. Basically says, people have attributed this sort of the dose makes the poison idea to him. His actual quote is that everything is a poison, and it's just a matter of how much you get, essentially how much you get of something that makes something poison. So the idea is that basically the idea of dose response. You start with no effect or some therapeutic effect, and eventually if you increase dose enough, you'll get to a toxic effect. I say unfortunate because this is led to basically this idea of just low ALs and no ALs, and this is all you need to know from toxicological studies. No AL is basically this point here where there's no statistically or biologically significant change. Most of the time, even though they talk about biologically significant, they really just mean statistically. And it's used as a point of departure for deriving toxicity values and how much we're allowed to be exposed to in drinking water and air, etc. And it's commonly viewed incorrectly. Probably, I would say maybe 75% of the SOT and probably 90% of those above a certain age view no ALs as an actual experimental dose threshold. So, which is obviously mathematically incorrect because you're just saying you can't tell the difference between the control. It doesn't mean that with more precision you wouldn't be able to actually see something. But that's the the way things have been. They take this no AL divided by 100, and then you say, well, you can be exposed to that amount and not worry. Before it was just 100. This was Lehman and Fitzhugh back in the 50s or 60s. Then they decided, well, we should divide the 100 into 10 and 10, one for animal to human, one for human variability. And then later, like, well, maybe we should divide each of those 10s into 3 and 3 for toxicokinetics and toxodynamics. This is all post hoc reasoning, right? There's no reason why these are 3 or this is 10 or this is 100. It's just because we happen to count in base 10 because we have 10 fingers.
And it's also always struck me as absurd that the 3 and 3 don't multiply up to 10.
Exactly. Does everyone know what a DABT is? Diplomat American Board of Toxicology. So a lot of professional toxicologists get these 4 letters after their name. So recently, one of my colleagues up in Canada said they should rename DABT divide arbitrarily by 10. So the last 25 years of risk assessment has really been focused on this idea of reducing uncertainty. So human relevance of experimental animal studies. This human relevance framework, can we throw out animal studies because we decided they're not relevant? So that was kind of the 90s. In the 2000s, it was about physiologically based pharmacokinetic models. So even though they've been around for many, many decades before that, this is when these models began to be used more routinely in risk assessment and to reduce, interspecies extrapolation, uncertainties, etc. So if you get the same dose, and even though you have the same dose, you might not have the same tissue concentration between humans and animals. And then in the 2010s, this idea is using systematic review to more objectively look across the body of literature in sort of a structured way, taking lessons from evidence-based medicine, because medical treatments often used to be just based on quasi-anecdotal data as to what works or what doesn't, and evidence-based medicine was trying to bring these systematic review approaches to make sure we have a rigorous evaluation of the whole body of evidence. And now that has become, sort of the new dogma in environmental risk assessment as well. But so uncertainty is only one half of the equation here. So David Threadgill, a colleague of mine at AM, likes to quote this guy Claudius Galenus or Galen of Pergamum, he actually really was sort of one of the fathers of medicine. This was many centuries before Paracelsus. And he basically was foreshadowing gene-environment interactions, saying that there's no external cause that is sufficient without predisposition of the body. Otherwise, the same cause would affect everyone, right? The same thing would affect everyone. So obviously, people have different systems, resistance, susceptibility. So this is came before Paracelsus, and so we should actually probably recognize we should first think about gene-environment interactions before we think about dose response. So how would we go about characterizing this variability? And there's really a lot of challenges in doing this with the tools that we generally have. So epidemiologic studies and clinical studies, they have really limited power to examine susceptibility and variability. And there's also the issue of generalizing from the cohorts that are studied to the general population, which is usually much more heterogeneous. This is why a lot of drugs, they don't fail to actually put them on the market, because then now you have millions of people rather than maybe even 500 or 1,000 people taking. Animal bioassays, they're almost always done in homogeneous experimental animals, so you're never going to get any information about variability from those. Toxicokinetic models, again, they're really, most of the models out there are just like a single model for a single species and not looking at the fact that people vary in their body composition, their enzyme activity, etc. Even in vitro, since the last 10 years, since toxicity testing in the 21st century, vast majority of these in vitro systems are in homogeneous systems with only, one genome, and after it's been processed, to make it a cell line, etc. like that, who knows what relation that had even to original human, let alone the whole population. Adverse outcome pathways, that's the a new buzzword for mode of action, but again, susceptibility and variability is not generally addressed there. So in the end, we have these risk characterizations and toxicity values that really just, let's just divide by 10 and assume that covers enough people. So the challenge really is, you start with, whether it's in silico, in vitro, or in vivo data, and you want to make predictions for a variable population. How do you actually make those inferences from these much more homogeneous systems? Now, obviously, for experimental work, you like homogeneous systems because you want to control everything, and so that from an experimental point of view, it's good to have a very homogeneous system. But in terms of making the trouble with that is the generalizability is much less the more homogeneous your system is. So, risk assessment is traditionally divided into hazard identification, dose response, risk characterization. And so I'm going to talk a little bit about each of these pieces of risk assessment as to some of the challenges and opportunities. So in terms of hazard identification, I think I've already mentioned this, in terms of a single strain, whether it's positive or negative findings, if you are finding them in only a single strain, you can't necessarily be sure that those generalize to a human population. For mechanisms, you can do knockout studies, probe one gene at a time, so that can be very slow in looking at, well, you might know there's variation in this one gene, but what about most of the traits are multifactorial, polygenetic, and also when you're using animals, it's difficult to distinguish what might be an interspecies versus an intraspecies susceptibility difference. For dose response, it's just assume that whatever strain you tested in is representative of the population, and then we just use these sort of tenfold factors. So in the last 5 years or so, there's been development of a number of population-based tools and approaches that have the potential to really get at this other dimension of variability. One is I'm going to talk about in sort of some in vivo models, and then talk about some in vitro models. And all these involve studying populations in the experimental setting, and it's not just to have more variability, but to have reproducible variability. So the fact that it's homogeneous in that every time we do the variability study, we're getting the same amount of variability. So in that sense, it is controlled, but it's a controlled variability. So hazard identification. So why should we use these models? the the hypothesis is that you might have different strains that are better or worse models for humans across the population. And so if you test a larger number of strains, you're more likely to sort of overlap with the range of human responses. So you reduce the chance of being unlucky and picking a poor model for humans, and then also you can potentially get some information about the range of variability in mice might actually be a surrogate for the range of variability in humans. So one of the resources that's been developed is what's called the collaborative cross. This was really a genetics problem. They took 8 founder strains, most of which were inbred, but some were outbred. And then each of these strains was crossed for many generations, and then they to sort of mix up their genomes. And then after a certain point, then they started to inbreed them again. So now you have hundreds of independent inbred strains. So each of them is homogeneous, but you have a panel of several hundred of them that can be a resource to examine population variability in a controlled manner. So one of the fascinating things about these animals is the amount of just baseline control variability, without any treatment that they exhibit. So how many people here actually like run or jog? Maybe about half of you. So mice, if you give them a treadmill in their cage, so they'll generally just run on it for a certain amount of time, during their waking period. So this is looking at 100 or so different strains. This is before they get to the inbred, but still it makes the same point. And this is the number of kilometers they run average overnight, while they're awake. These are real kilometers. These are not mice-scaled kilometers, right? So these strains are running like 20 kilometers every night. This is an average over several nights. So these are like the marathon, the people who are addicted to marathons, right? And then there's some that you can't even see how small the amount that they run is every day. So these are the couch potato mice down here. This is like 1 pixel high, this last mouse over here. And there's 2 things. One is this extreme range of just ad libitum running, right? Some people really don't like to do it, or some mice and people, and some just love to do it. And the range is actually wider than the individual strains themselves. So you wouldn't have guessed that. This top here is twice as much as as one of the 8 original founder strains. Now the lazy mouse, there's some that are even lazier, the original ones. But this is several, 2 is here, so tenfold is from here to here, so it's more than tenfold range of distance running per night. Another example with an actual treatment is Ebola. So Ebola, there's been these outbreaks that have been in the news in the last few years. And traditionally it was thought that mice were poor models for Ebola because they didn't get hemorrhage and have these, sort of classic Ebola symptoms. But when they tested in the collaborative cross, they get a range of phenotypes. There are these ones that are resistant. This is the dark blue over here. And then there are ones here that are in the pink that are partially resistant, and then ones with like severe. These are the ones with the classic. It's lethal and they bleed out essentially, right? And so you have this range of phenotypes from across these different strains. And interestingly, because we've been traditionally probably testing here where we would basically miss the hazard. So if we were testing some other stressor and we were testing the resistant strains, then we would miss the fact that there might be a human hazard. And then interestingly, this isn't super quantitative, right? Because it's hard to do the exact diagnosis, particularly in humans. But there's about 70% lethality in humans in one of the more recent outbreaks. 30 to 50% are hemorrhagic, and so that it's not too different than what we see across these different mice strains. So this suggests that looking at an entire population of mice can actually be very informative as to human hazards. So there's some older studies that used, before the collaborative cross, that used sort of inbred panels of mouse strains. And this is an example with acetaminophen, both a human and an experimental mouse study. So this is the range of, this is serum ALT changes across different humans, and some people have no change essentially, and some have like a 7-8-fold change in ALT from taking Tylenol at the prescribed maximum dose. Now these two sets are different doses, and they're not exactly comparable. So the lower dose here, again, there's a range of amongst these 6 strains, and then at higher dose, there's some of these mice that even at a higher dose don't show any ALT changes. They don't seem to be getting injured in the liver, and then some with very, very obviously very, very large differences. So if we're testing new compounds and we're just happen to be picking resistant strains, we're going to miss a human hazard. And then the other thing is that these distributions do sort of overlap. So basically, for hazard identification, even identifying things that might be harmful or not to humans, these genetically diverse mice can potentially provide a better signal for what might happen in the human population. So the opportunities then, higher probability of overlapping with humans, and potentially directly informing how wide that range of human variability is. Challenges, the data analysis statistical modeling approaches have been developed, in laboratory experiments, not accounting for this sort of population variability component. So there's some, the methods exist. The stats people have, invented a long ago, but they just haven't been used. And so you can't just use the same analysis you've always done in analyzing these type of data. And then there's sort of the cost benefit. So what's the, on a fixed, there's always resource constraints, right? So on a fixed budget, when should you use a population model versus a single strain model in terms of power to detect? So some of those things, need to be, investigated and worked out. And without more case examples, it's hard to know what that balance is actually going to be.
Basically, with a model that had variability, and wouldn't the test sample typically be larger? Would you have to commit to a larger number of animals to embark on a study like that? Kept the study the same size, use variability at the bad practice?
Yeah, just I guess so. With a single strain, it's more binary. Like if you just happen to pick a bad model, like none of your results mean anything, right? And whereas, so you're spreading your bets across a larger number, but the individual power for each individual strain might be less. But you're testing a wider range of doing more tests essentially. So so for fixed power on a specific strain, you would obviously need to have larger amounts. But
I was thinking of statistical power to answer the question whether if the strain has an adverse response. But you're thinking of the power to extrapolate to including it to all of the humans, right? So
right.
including the fact that the strain may miss entirely the effective.
Right.
Make good data on that strain, but it may be irrelevant.
So I think that's sort of a bigger picture trade-off, I guess. So that's why I think without more case studies as to how much are we really missing with single strains, it's hard to know what this trade-off would be.
And a priori, you have no idea.
Right.
For people who do this in practice, would they use typically a larger sample set?
Well, so there's also some technical issues. It's hard, like some of these strains don't breed that well, and so you end up not getting that many, it's for the again, on a fixed budget, it's not like you can just buy them from Charles River, right? So it can be more expensive even to get one mouse, for some of these strains. And another, this is more related to dose response, but people have argued that if you're committed to doing dose response modeling, you should actually have more doses and less animals per dose. Because if you're going to fit it with a curve, it's actually better to have more And the curve fitting essentially accounts for the you get better power to spreading, having fewer replicates, and more doses. So in a way, you could think of maybe that's analogous here as well. Maybe you get more power by having lots of strains, even if it's only a single mouse per strain, than having lots of replicates per strain.
When you come to interpreting that data, you say you get an adverse effect for one strain.
One mouse.
You'd have to ask yourself if this is bad data or it'd be hard to.
Well, so that's the advantage of these inbred models, that you can actually go back, and then the ones that seem to have an effect, you could go back to the same strain and do a replicate study on those particular strains to see if you actually if it was just noise.
Find out whether the mouse just keeled over.
So a second area for risk assessment is trying to investigate mechanisms of toxicity and susceptibility, and this is really where the sort of gene-environment interactions you can explore with with these population-based resources, because one advantage of this designed population is not like the human population, which is very lumpy in terms of the fractions of various variants there are. These were designed to be very balanced. So each variant is has a each of these original founder strains, on average, 1/8 of them is in each mouse, right? It's just it's all just mixed up. So you can statistical geneticists tell me that this means you have higher power. You need a smaller sample size to get the same power to find alleles that have a low size actually. And so you could look at those same pathways that seem to be genetically affected as well as environmentally affected, and kind of parse out some mechanistic information from that. And so one of the proofs of principle is again with acetaminophen. So this is liver toxicity. This is the liver toxicity in humans, and then in mice across different strains. And then they essentially did a GWAS in these mice, and they found several that it's too small to see like this 4 here. So this is like 3.5 strength of association. And one of these was the CD44 candidate gene, susceptibility gene, and then they went back to a cohort and looked at this gene in the human cohort, and found that indeed people with different variants have different responses to acetaminophen. So this was confirmed in 2 independent cohorts, and that provided insights into the mechanism. The CD44 status actually is related to after injury, whether it goes to recovery or whether it goes to apoptosis and inflammation. And so there's actually, no one imagined that this pathway was actually involved in acetaminophen toxicity. People thought it was just sort of a classic, cell killing kind of thing. But really, the difference between people who recover and people who don't is related to the CD44 status. So that's sort of proof of principle of how you could use this to do something, to basically identify potential mechanisms through this identifying susceptibility. So opportunities here are, the genetic basis for susceptibility, being able to make confirmation, like hypothesis generation almost, like in these experimental systems that you can then go target in humans. And then maybe in the future some sort of personalized risk assessment for each person's different variants. Now, circle back to that at the end in terms of issues for risk characterization. Challenges, of course, most things are not single gene susceptibilities, and of course there's all these non-genetic sources of variability from our broader environment or epigenome that could be influential. So finally, in terms of dose response, so why should we use these population-based models? Here again is just the same thing flipped on its side. You have a point of departure, you divide by 10 twice, and then you have some toxicity value that we presume or assume is protective of the population. So on this end here, these typical strains might not be representative or even relevant to humans. So we don't know whether these factors of 10 are really appropriate or not. So using these population-based models, we could actually potentially get a better estimate of what the average animal is versus, and then they're either use models or things like allometric scaling to estimate what the average human might be from that. We can also use them, these models, to better understand toxicokinetic and toxodynamic variability, because we have experimental systems to explore the variability, and then we can use models then to fit that how much variability we observe. And ultimately, the idea is to actually characterize what fraction of the population might be we're actually going to be protective of. Since I'll return to this at the end, you can't guarantee 100% safety to 100% of the population. There's some amount of risk tolerance that we have, but right now it's completely untransparent as to how conservative or how protective we be. So I'm going to focus now here on this 3 and 3 for human variability. Can we better estimate the population variability first on toxicokinetics and then toxodynamics? So PBPK modeling is essentially the go-to approach here. This isn't really new, but it might be new to folks here. In terms of using Monte Carlo simulation, various Bayesian approaches to try to estimate how much variability in sort of internal dose there is across the population. But then I'm going to show how some experimental models could actually augment some of that work as well. So the basic idea behind population PBPK modeling is that first you have this model, but instead of fixing the size of the lung, the blood flow to the liver, etc., you actually allow those to vary, both in terms of physiological parameters, physical chemical parameters like how much they partition to various tissues, and then biochemical parameters like the Vmax, Km, and things like that, metabolism in the liver. That's going to be allowed to vary. So how many of you have heard of Simcyp? So they're a firm out of UK that has developed sort of mostly for pharmaceutical uses, developed approaches. They basically have fixed these distributions based on data that they've collected, and then provide pharmaceutical firms with ideas of like how much variability in dosing would you expect for particular chemicals. In Bayesian approaches, these are not sort of fixed, postulated distributions, but these are conditioned on data that you have. But either way, the ultimate output is that you have a distribution for what the concentration of a particular metabolite in a particular tissue or is across the population. So there's a lot of this variability is simply due to physiology. For instance, a lot of chemicals are with a very large first pass. For instance, it's going to be basically limited by blood flow to the liver, and so that's fortunate because we can collect a lot of basically independent data that's not chemical specific to inform this type of modeling. We also have a good idea based on clinical data from drugs as to what the possible range of toxicokinetic variability is. And this is just work by Dale Haddis at Clark University. They collected over a decade or more. So if you look at, this is like for AUCs, if you look across different chemicals, some pharmaceuticals, for instance, people are fairly narrow in how variable they are. And some chemicals, there's a lot of variability. It just depends on what enzymes, for instance, are mediating metabolism. And so this distribution here is the ratio between sort of a median person and a 1% more sensitive person. Like how much more dose would the more sensitive person get relative to the median person. And this could, depending on the chemical, some are like 1.4-fold, a pretty small range, and some might be almost 10-fold in terms of how much additional internal dose they get. So this is great so there's a wide range across chemicals, and so for any particular chemical, you might be a low variability chemical or high variability one. And in general, in the past, we've needed actual human data in order to figure out which ones are low variability chemicals and which ones are high variability ones. And this has been done for like trichloroethylene and some other chemicals that have a high profile. But the question is, can we maybe have an experimental model that can mimic this human variability? So this is where the work that I'd done previously for trichloroethylene with EVA was useful as sort of, we have actual data from humans, and then if we do the same thing in a population of mice, do we get something that could serve as a surrogate for human variability? So for the human model, and then this depends on which internal dose metric you're using. So if you're just talking about oxidation, then it's very little variability across humans. But if you're talking about conjugation with GSH, you're getting 7-fold variation, and this is the 95th to 50th percentile range in terms of variability. And then because data are imperfect, so we have confidence intervals on each of these. So the question is, and also there's only about 50 individuals that have been tested for TC over the course of probably 60 years of research. Most of these are in an occupational context. They wanted to look at biomarkers of exposure for occupational hygiene, for industrial hygiene purposes, and so it was collected from many different studies. So what happens if we do this in various strains of mice? So this is different metabolites. This is the B6C3F1 strain that's typically used for toxicity testing. And then these are two other strains out of a panel of 17 strains that were used. As you can see, there's definitely variability, particularly for some of these for this GSH conjugation metabolite here, DCBG. The B6C3F1 strain is quite a bit, and these two have very little. But even for some of these other dose metrics, there's definitely variability that is detectable across the population. So we put this into the model again, and then we can compare, well, were the predictions from this population of mice anywhere near the predictions that we got from actual human data? And this is another one of these cases where after I did this, I was like, wow, that actually worked. So, and it basically reproduced within these confidence intervals the same results as in the human variability data. So for just oxidation, very little variability. For GSH conjugation, about a 7-fold variability. And if you look at these confidence intervals, they all completely overlap. Even just rounding to one significant digit, they're the same, essentially. So this gives us some hope that using mouse populations, at least particularly here in terms of toxicokinetics, it might be as long as you have a good mechanistic model in terms of this PBPK model here as to how the metabolism pathways and the function, that you can actually get a pretty good surrogate for human variability from these experimental systems. And given that we're probably not going to be dosing humans with these chemicals and getting toxicokinetic data from them for industrial chemicals in the future, this would be essentially the only approach to get actual empirical data on variability.
Did you do this estimate? I think it's the DCVC that is the actual kidney carcinogen.
Mm-hmm.
Did you do that estimate? It looked like the variability was less for that metabolite. Did you do the estimate for that metabolite?
We did, but because in the original TC assessment we actually used this total fraction of GSH conjugate. And actually in the human data it wasn't that different here. So I don't recall exactly for that particular metabolite in the kidney. A lot of them were also got to be non-detect, so that it gets difficult because it goes away pretty quickly.
What is the biochemical significance of TCE conjugated with GSH? What is that?
That's the man.
So it's conjugated with GSH. One of the chlorines goes away, so it's dichlorovinyl glutathione, and then it gets processed to a cysteine conjugate, and then so those two are like Ames positive in certain assays. They then get bioactivated into reactive metabolites, which can either damage DNA or cause cell damage. So whereas the oxidative metabolites like trichloroacetic acid, those are stable circulating metabolites that don't act on don't cause direct damage in tissues. So that's why there is a lot of interest in this. And then because a lot of this bioactivation occurs in the kidney, and kidney cancer is one of the targets that's been found in human studies as well as in cell rat studies, kidney cancer. So that was the interest in this GSH conjugation.
That looks like a reductive dechlorination or something like that.
Yeah. So usually you think of GSH as a detoxification pathway, but in this case it actually leads to formation of reactive metabolites. So the next step is to do this. So that was with these inbred panels of mouse strains. The next step, we actually have done some experiments with the collaborative cross, and interestingly, toxikinetics is a complex trait, even though people think, oh, it's just the CYP2E1 or whatever. So this is the variation of CYP2E1 in the liver across these different strains, about 45 of them. As you can see, again, very, very large amount of variation. And then these two down here, which have small amounts of CYP2E1, one of them has very little TCA, and one of them has a lot of TCA. So this is not intuitive as to, you would think if there's more oxidation, then you could have these two would be up here with the more TCA. But it's a complex metabolism pathways. TCA is formed not just from CYP2E1, but also through trifluoroethanol, through the aldehyde dehydrogenase. So these are also different tissues. So you can't make predictions. Even if it might seem reasonable, qualitatively, when you look at this in detail, it actually might not pan out. So I like seeing the ultimate empirical data as to what the actual variation is, and not necessarily just making leaps of faith based on mechanistic hypotheses. So this has also been used for in terms of, again, the sensitive idea of just based on in vitro data from various human hepatocyte data, essentially. And then the idea here is to try to, here it's not so much the general population, but looking at different subpopulations and kind of piecing together different SIPs in the liver. So you're basically creating livers for neonates, livers for Asian, livers for Northern Europeans, etc. They have different distributions of these different SIPs, and then basically make predictions for subpopulations. And so some of this this is work by Barbara Wetmore, who's now at UVA. So the next question is toxodynamic variability. And again, based on work from Dale, mostly on pharmaceuticals, there is some idea as to how much toxodynamic variability there is in the population based on clinical data, looking at essentially the relationship between serum concentration and some toxic effect. And this range, 3 is actually around the median, but some toxodynamic effects are much highly variable and some are not. But there's virtually no examples for chemical-specific estimates for environmental chemicals as to how much toxodynamic variability is. So what we did is, this was based on this work that one of Yvonne Russin's graduate students did in collaboration with NCATS and NIEHS using the 1,000 Genomes project, where they took lymphoblastoid cells derived from 1,000 different individuals from across the globe in terms of different populations, and then did cytotoxicity screening of about 170 different chemicals. So this was a massive experiment, right? In 1,000 individuals by 170 chemicals by concentration response with replicates, to look at batch effects. So Noor Abdo did a huge amount of work to get this done. And that's why we had to partner with NCATS, because they have the robots and things like that. They can do some of this work. And we picked compounds they picked, this was while I was still at EPA that they did the experiments, that were structurally diverse. And this is like PCA with various, these chemochromatic kind of things. And, they actually are a fairly good representation of, the known sort of chemical space for relevance to environmental chemicals. And so what you end up with is, graphs like this, where this is cytotoxicity, cells dying, and some chemicals, they all die around the same concentration. And then some chemicals, there's very sensitive cell lines and very resistant cell lines. So depending on the chemical, you could, this is cadmium chloride here, only about a 2-fold range. And then some of these mercury compounds have a very wide range, of response across these 1,000 individuals. So, does this, so this is cytotoxicity in lymphoblastoid cells, right? So does this have anything to do with actual human responses? And so we compared this with the work that Dale Haddis had done looking at actual human clinical data in terms of the distribution across chemicals. So you have chemicals with low variability, chemicals with high variability. These are different sets of chemicals as well, but just looking, if we think of the chemical universe and these as samples from the chemical universe, do they give even the same ballpark range? And there's another one of these cases. Oh my goodness, it works. So you have the red line here is the in vitro data, and then the black line is the in vivo data. So this is the cumulative distribution of how many chemicals have variability of up to twofold, tenfold. This is 100-fold up here. There's nothing in 100-fold. But this toxodynamic variability factor, is about threefold on average. So the median's about threefold, and then the range across the different chemicals is pretty wide from just less than 2 to greater than 10. So even though this is you wouldn't expect an exact correspondence, right? These are different chemicals, and these estimates from in vivo are difficult to estimate as well. But definitely in the same ballpark. And actually, when I redid this using a Bayesian method, the in vitro one actually went smaller a bit because there's some noise that I was able to take out using a Bayesian method. So it isn't quite as good as this original paper was when I redid it. So that's the problem if you investigate after it works. If you keep on investigating it, actually sometimes it doesn't work after you look into it more detail.
Isn't that 3? I mean, is that sort of justification for the 3 in your last split out of the table? Isn't that a toxodynamic?
Right. It's just coincidence, right? So
it's not a conservative value. It's kind of an average value.
Right. So that's the difference. So people say, "Oh, 3 is fine." Well, but people think of the 3 and 3 as conservative, right? That it's no more than 10, but really we're saying 3 is typical amount of variation, and not a conservative value, which would be higher. So the next step is really to not use lymphoblastoid cells and to look at different cell types. And so some work that in Ron Russo's lab looking at basically IPCS-based models from hepatocytes, cardiomyocytes, neurons, etc. The difficulty is getting the variability component here because it's not, cardiomyocytes are fairly easy to differentiate, and these are all cells that we actually purchase from Cellular Dynamics and the CDI. So we're not doing differentiation in our own laboratory, but they guarantee like the differentiation, and they do quality control and everything. But potentially, if we're able to have panels of individuals across different cell types, you could actually look at more phenotypically interesting or relevant outcomes, and again, be able to better estimate what this taxodynamic variability might be, which is very difficult to do in vivo in humans. So, taking cardiomyocytes as the example, so this is an EPA grant to originally look at 100 different individual donors and 140 different chemicals again. This is the set of 27. I think we're only going to get up to maybe 40 or something because EPA wants us to focus on testing more chemicals rather than more individuals. But this is just the beats per minute, so their baseline heart rate. So some people have slower heart rates and some people have faster heart rates. And this is just cardiomyocytes. So these are unpaced, right? So this is their kind of spontaneous beating frequency, which isn't the same as people's actual beating frequency. But the fact that there is this variation, and then they looked at it, a year later with a different batch from the same donors, and they basically get the same. The high ones are still high, the ones are still low. So this isn't just, random variation. These are actually reproducible variability over time. And then they also have different phenotypes in terms of if you could treat them. So this is a toxic drug, sotalol, that causes QT prolongation. So for this really fast beating guy, he or she is just beats a little bit faster. Here, this moderately beating person, they exhibit this shoulder, which is a delayed action potential, a delayed repolarization. So this is the in vitro phenotype corresponding to severe QT prolongation. And then this person down here, who's sort of a slow beater, they actually almost get a small second beat before the main beat goes on. So this person is getting fibrillation essentially. So you're getting variation in response along with variation in baseline. So this is really promising in terms of really looking at chemical specific effects and the variability across the population of those. I think in interest of time, I'll skip some of this other work that's been done in vivo in humans. This is for a paper by Jeff French in terms of benzene. These are micronuclei. This is using another population called diversity outbred. The main point here is just that the standard mouse is less sensitive than the average of this population. So, for this particular endpoint, B63F1 mice are resistant. Now for TCE, B63F1 tended to be on the less resistant side. So depending on the compounding endpoint, your typical mouse might be more resistant or more sensitive. And then for TCE, we actually did, this is crazy to do in an academic lab, right? We did a 90-day study in 50 strains. I think it was 50. It was 20 or 50 strains. No, it wasn't 50. I think it was 20 strains of mice. So I'm glad I wasn't in that. But, so anyway, and so the preliminary results, again, there's a lot of inter-strain variability even at baseline, liver body weight ratios, hemoglobin, body weight certainly, kidney weights. And then, we're doing some analysis of data, so this is one of the stay tuned things. So the advantages of using these models for dose response is really about you're testing population dose response, you're getting better idea as to this the amount of variability across the population, and it seems like some of these experimental systems might not be bad surrogates for the actual human in vivo situation. So these approaches are all very quantitative, using population-based statistical models driven by data, and it seems odd to after doing that just change your safety factor from 3 to 5. So use a NOAL and then go on to calculate an RFD that way. So really what I'm advocating for is that this is a perfect way to integrate a more probabilistic approach to actual toxicity value generation. And that way, it basically replacing each of these uncertainty factors and the point of departure with the distribution that, in the absence of data, can be based on historical data, like the stuff that Dale Haddis has developed. But with chemical-specific data, again, you have a distribution, not just for the uncertainty, but also for the variability. So this is work done with the WHO work group. And basically, we're trying to predict the dose response, the population of dose response curves in the humans based on experimental animal data, and accounting for the fact that we have uncertainty as to the animal to human extrapolation. We have variability, but we're also uncertain about how big the variability is. And I like to say that this is making the definition of the reference dose more precise. So this is on the left is the typical definition of the reference dose. These are all completely vague terms, right? Sensitive subgroups likely to be without appreciable risk of deleterious effects. What is a deleterious effect? What is appreciable risk? What does likely mean? The idea is to make all these, force people to assign a number of each of these. So we mean a particular statistical confidence limit. We're talking about a specific fraction of the population with a potential residual risk. And then we have this deleterious effect, we actually said, well, we mean an effect of this magnitude for this particular endpoint. And so really this reference dose is really some, diplomatic, vague concept that is really an approximation to this more precisely defined, we're calling a probabilistic reference dose. Yep.
Yeah, this is appealing to scientists, but you can imagine that other one much more appealing to the communication to the public.
That is a challenge.
If you name the fraction of people who are going to get a disease, and you name what the disease is, you say, well,
This is the best we can do.
That's pretty good.
Yeah.
Put it into law. You agree.
But this is just sweeping it all under the rug. It's about forcing risk managers to make tough choices that they always wanted to blame on the risk assessors before,
Or the
or the scientists. Yeah. So there's definitely opportunities for this, but as you allude to, the challenge is engagement and communication amongst risk assessors, risk managers, researchers, etc. And I wanted to end with this focus on challenges to risk characterization. We essentially need to acknowledge that what we call safe exposures are not risk-free. It's not 100% certainty. We're not saying we're going to protect every single human being on the planet. And but right now we're just sort of putting our head in the sands and just pretending to no la 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 la. And so I actually did an analysis and saying, well, what is the actual risk at the RFD if we take these distributions and use them? And this is actually a poster at SOT in a few weeks. So I applied this framework to 1,500 different endpoints for 600 different chemicals and calculated, well, what is the confidence bound on the incidence in the population for that effect that they use for the RFD. So this here is the upper 95th percentile, this is the median estimate, and this is the lower 95th percentile. And this is a hazard quotient of 1, so exposure at the reference dose. As you can see, there's a number of chemicals for which we're talking about as the upper confidence bound as a fairly large number, fraction of the population that we can't rule out might have an effect from being exposed at the reference dose. Now the central estimate is all pretty small, just a few little outliers here. This was like a dog study that they only had a certainty factor of 3 or something. And there might have been good reasons for that. And then lower confidence bound obviously it's tiny. But the other thing that came out from looking at this is that these endpoints vary hugely in terms of their severity. Right? We're pretending all RFDs are the same, but some of these are like small changes in clinical chemistry, and some of them are like, this is mortality that they're using to estimate an RFD from. So, a 1% change, 1% of the population having clinical chemistry changes is very different than 1% of the population undergoing mortality, right? So, so it really confirms that the RFD is not risk-free. These are very wide confidence bounds because this is really the best we can do. But if we're going to be honest as to what the data are saying and not saying, then this is really a more scientifically honest approach, even if it makes the risk managers uncomfortable. So another challenge is that if we actually probe this too much, we might actually be able to identify who might be remaining at risk. If we know in terms of the variability, what either genetics or other factors are causing them to be on that tail of the distribution, that doesn't really align with how our policies are designed, which are that it's risk, but it's unidentifiable. Some people are just unlucky, right? There is a precedent. Everyone can go online and calculate their cardiovascular risk profile based on their blood pressure, clinical serum cholesterol levels, etc. And what does that mean if you send your thing off to 23andMe, they'll culture iPCS cells, and they can test that your liver is really susceptible to this chemical, and or what does that mean in terms of like society, in terms of risk management, in terms of policy? Now there's sort of an undiscovered country here. So there's a lot of interesting challenges in terms of if we actually delve into this data, what it might mean for policy and decision making.
Even for the cardiovascular risk calculator, there's a huge controversy about that.
Right.
Whether 50% of the population is going to be taking statins versus 20%.
Yeah. Mm-hmm. Right. I mean, there are some doctors who say we should put statins in the drinking water because, that it will help everyone.
So actually drink water.
Yeah.
So to summarize, hazard identification, we're talking about models that are more likely to have some overlap with human relevance, and also identifying potential mechanisms. We've talked extensively about improving dose response, talked about the challenges to communication and policy, And I wanted to just end briefly, since this is the Superfund program, we are incorporating some of these into our Superfund research program. So for those of you who don't know, our program is focused on exposure to mixtures after a disaster. That a hurricane comes in and dumps a bunch of sludge from the Galveston Bay into people's yards. What's the risk from that? Have we just made all of Houston a Superfund site? Especially when you have 100-year floods every year for 3 years in a row.
You're assuming it wasn't a Superfund site beforehand.
Right. Yeah. So in a number of our projects, we're actually incorporating some of these, both the in vivo and in vitro approaches, and then me as part of decision science and translation core, trying to also bring in some of the in silico approaches to basically look at this population variability element as well as we move on with our Superfund research. So it isn't just theoretical. We actually try to apply this in a way that we could potentially translate to to the communities. Okay, and that's my last official. Sorry, I went over a little bit.
Thanks for excellent talk. Any additional questions? We sort of threw questions at you during the time you were speaking. So, Sue.
So most of the toxicity testing in big farm is done in rats. Are there collaborative cross rat models coming on, or did the trend or the move toward using mice?
There are some resources in rats in terms of genetic variability. Nothing like the statistical genetics designed population with the collaborative cross. I don't know the reasons for that. I think the mice are easier to read or easier to do something with. And pharma is actually very resistant to using these population-based models because
They don't want to know.
essentially, because if they can get it to be approved, if FDA is not asking for it, then they don't want to look under the rock and see what they're. Or if they do it, they're going to keep it inside their bubble, right, and not tell anyone.
They can only do that if they don't know.
So Alison Harrell, was one of Yvonne's graduate students, she's now at the NTP, but she was, after her acetaminophen work, she was trying to convince, pharma to take on some of these models, and there's just really big resistance to even considering doing any testing in them. Eventually, because originally she was going to go kind of a more clinical route in terms of her career, but now she's at NTP, where they do care about this. So.
Any other questions? Comments?
So it looks like, I'm not from that field, so bear with me a second. Looks like this is a manual application to fuzzy logic, because it's not a binary situation. Do you know of any attempts use fuzzy logic for risk assessment?
So the stuff that I'm doing is more traditional statistical distributions and combining things by Monte Carlo and things like that. But there is this guy, Scott Ferson, who's in the UK somewhere, who does the fuzzy logic stuff in terms of addressing uncertainty. So his argument is that you don't know what the correlation structure is, and even if you assume, a multivariate normal, it's much just the, and if you assume completely unknown correlation coefficient, it's still constraining as to what those relationships might be, just by the very fact that you're using a parameterized distribution. And so he has this thing called p-bounds, I don't know if you've heard of those, which is basically taking possibility theory with fuzzy logic to and applying it to actual cumulative distributions. So it's not your uncertainty in a particular number, it's your possibility, your fuzziness of a single number, but it's the fuzziness of what a cumulative distribution looks like. And he had, there's mathematical ways to combine them. So essentially, instead of, the possibility bounds, are like that, right? fuzzy logic. So he's basically taking each of the, so he's saying, well, basically doing this on quantiles. So you have a possibility for the 50th percentile, a possibility for the 55th percentile, and a possibility for the 25th percentile, that looks like that. And then so then you get a cumulative distribution that's kind of a box. And then he says, you can combine these, probably, possibilistically, I guess you would call it.
So he's kind of discretizing his range and using at each discrete section a fuzzy, as if it's a fuzzy.
Right, so each quantile is a fuzzy number. And then the problem with this is that if you get more than a few parameters, you end up with a square box, essentially. Right?
All possibilities.
All possibilities from the minimum to the maximum. So there's maybe that's actually the state of nature, that we actually don't know, anything between 0 to infinity. But
But we're paying for it.
right.
All right, thank you very much.
I have a few cards if people want.